In this section, you will get to know of the regulation of heart pumping through ventricular function curves, control of the heart by the sympathetic and parasympathetic nerves, and mechanisms of excitation of the heart by the sympathetic nerves. So let's start with ventricular function curves. One of the best ways to express the functional ability of the ventricles to pump blood is by ventricular function curves. This is left ventricular stroke work curve, and this is right ventricular stroke work. These are types of ventricular function curve called the stroke work output curve. Atrial pressure for each side of the heart increases, stroke work output for that side increases until it reaches the limit of the ventricle's pumping ability. These are actually left and right ventricular function curves recorded from dogs, depicting ventricular stroke work output as a function of left and right mean atrial pressures. Another type of ventricular function curve called the ventricular volume output curve. The two curves of this figure represent function of the two ventricles of the human heart based on data extrapolated from experimental animal studies. As the right and left atrial pressures increase, the respective ventricular volume outputs per minute also increase. Thus, ventricular function curves are another way of expressing the Frank-Starling mechanism of the heart. That is, as the ventricles fill in response to higher atrial pressures, each ventricular volume and strength of cardiac muscle contraction increase, causing the heart to pump increased quantities of blood into the arteries. Now, let's discuss the control of the heart by the sympathetic and parasympathetic nerves. The vagus nerves to the heart are parasympathetic nerves. This is of the or atrioventricular node. This is SA or sinoatrial node. This is sympathetic chain. The pumping effectiveness of the heart also is controlled by the sympathetic and parasympathetic or vagus nerves, which abundantly supply the heart. For given levels of atrial pressure, the amount of blood pumped each minute, called as cardiac output, often can be increased more than 100% by sympathetic stimulation. By contrast, the output can be decreased to almost zero by vagal or parasympathetic stimulation. Now, moving towards mechanisms of excitation of the heart by the sympathetic nerves. Strong sympathetic stimulation can increase the heart rate in young adult humans from the normal rate of 70 beats per minute up to 180 to 200 beats per minute, and rarely even 250 beats per minute. Also, sympathetic stimulation may double the force of heart contraction, thereby increasing the volume of blood pumped and increasing the ejection pressure. Thus, sympathetic stimulation often can increase the maximum cardiac output as much as twofold to threefold, in addition to the increased output caused by the Frank-Starling mechanism. Conversely, inhibition of the sympathetic nerves to the heart can decrease cardiac pumping to a moderate extent. Under normal conditions, the sympathetic nerve fibers to the heart discharge continuously at a slow rate that maintains pumping at about 30% above that with no sympathetic stimulation. Therefore, when sympathetic nervous system activity is depressed below normal, both the heart rate and strength of ventricular muscle contraction decrease, thereby decreasing the level of cardiac pumping by as much as 30% below normal. Now let's discuss the parasympathetic, or vagal stimulation. Well, parasympathetic, or vagal stimulation, reduces heart rate and strength of contraction. Strong stimulation of the parasympathetic nerve fibers in the vagus nerves to the heart can stop the heartbeat for a few seconds. But then the heart usually escapes and beats at a rate of 20 to 40 beats per minute, as long as the parasympathetic stimulation continues. In addition, strong vagal stimulation can decrease the strength of heart muscle contraction by 20% to 30%. The vagal fibers are distributed mainly to the atria and not much to the ventricles, where the power contraction of the heart occurs. This distribution explains why the effect of vagal stimulation is mainly to decrease the heart rate rather than to decrease greatly the strength of heart contraction. Nevertheless, the great decrease in heart rate combined with a slight decrease in heart contraction strength can decrease ventricular pumping by 50% or more. Now let's discuss the effect of sympathetic or parasympathetic stimulation on a cardiac function curve. There are 4 cardiac function curves. This curve is for maximum sympathetic stimulation. This curve is for normal sympathetic stimulation. This curve is for zero sympathetic stimulation. This curve is for parasympathetic stimulation. They represent function of the entire heart rather than that of a single ventricle. They show the relationship between right atrial pressure at the input of the right heart and cardiac output from the left ventricle into the aorta. These curves demonstrate that at any given right atrial pressure, the cardiac output increases during increased sympathetic stimulation and decreases during increased parasympathetic stimulation. These changes in output caused by autonomic nervous system stimulation result from changes in heart rate and from changes in contractile strength of the heart. This was all about this section. In our next section, we will be discussing the effect of excess or deficiency of different ions on cardiac muscles.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.